All right, welcome to the first lecture of second semester. This is going to be section 5.2, or I'm sorry, 5.1, and this will be our basically our fifth unit in the throughout the whole year. So we're starting with DNA this year. What we're going to do here in this lecture is go over the history of DNA and then basically what it looks like. And then next lecture we'll talk about replication and how DNA is copied, which we did review in mitosis, but we'll review in more detail now. So let's get into the history. First thing I want to learn about DNA is how it was actually discovered. There were a few scientists that were involved in the discovery of DNA, one of them being Griffith. And Griffith actually was setting out to see how bacteria made people sick. He knew that there were certain kinds of pneumonia that killed mice and certain kinds that didn't, but he really wanted to figure out why they were getting sick and ultimately dying. So what he was doing was he was taking disease-causing pneumonia, disease-causing pneumonia, and harmless bacteria, really, which didn't cause disease, and injecting those into mice. The disease-causing bacteria ultimately killed the mice, but the harmless bacteria didn't. And what he discovered through doing that is that there was something that he could destroy, there was a different kind of bacteria, different types of bacteria that were all classified the same way, but there were ultimately different types and what was in them. So I'm going to show you this picture actually because I think it does a better job of showing you what occurred. But we can go back to the words here or the notes here in a minute. So this was basically Griffith's experiment. He was taking the disease-causing bacteria, injecting those into mice, and they were dying. So this is actually his control. Okay, the disease-causing bacteria were doing what they're supposed to. Harmless bacteria were being injected into the mice, and they were living. So again, the bacteria were the same, but there were two different types of the bacteria: the pneumonia, the disease-causing pneumonia, and the harmless pneumonia. Okay, so then he did a bunch of different trials on the bacteria to see what he could do to them and see how it affected the mice. So in one instance, he took the disease-causing bacteria and he heated it up, okay, using a flame. After doing that, he grew them and injected them into the mice, and after doing that, the mouse lived. So he discovered that he could really harm the bacteria by heating them up, and that would cause the mouse to live. Nothing would really happen to the mouse. But the second trial he did was involving the heat again. So he took the disease-causing bacteria and heated them up, and then he added harmless bacteria to the mixture. So there were both of these in the same container. He then injected that into the mouse, and the mouse died. And when he dissected the mouse, he discovered that there were disease-causing bacteria in there, even though he had killed them previously. So what he had ultimately discovered here, going back to the first slide, is transformation, and that's the note here at the bottom. Transformation is the process by which a piece of DNA in a bacterial cell gets changed from one bacteria to another. So basically, the disease-causing bacteria and the safe bacteria, so the disease-causing and the safe bacteria, can swap DNA. And if they swap DNA, they can do different things, whatever the DNA tells them to do, whether it's kill the mouse or allow the mouse to live. So that's what Griffith discovered, and Griffith was really then discovering here that information could be traded between bacteria, which was unusual. Other scientists that led to the discovery of DNA were Avery, who discovered that nucleic acids, discovered that the nucleic acid of DNA stored the information. But as Avery made his discoveries, people were still a little skeptical because Avery and Griffith were the only two that had worked on this experiment. The ones that really set it in stone were Hershey and Chase. And Hershey and Chase did this experiment where they basically highlighted the DNA in a virus and watched the virus transmit the DNA into the bacterial cell. And then, after it was done, they went back in and looked at the bacteria to see if the highlighted DNA made it into the bacterial cell. Because once the DNA got in there, those are the instructions to make more viruses. So Hershey and Chase did this, and they discovered that the genetic material of the bacteria And the virus, the bacteriophage, is just a virus that infects bacteria. The genetic material was DNA, and it wasn't protein. Okay, protein did not carry information. It was actually the DNA that carried the information. So those were the scientists that led to the discovery of DNA, and they were very influential in their time. So after this, at this point, then Watson and Crick come along, and they discover what it looks like. So just to review what we've already kind of learned earlier in the year, the DNA molecule is made up of nucleotides, and the nucleotide contains 3 segments. The first segment is the sugar, that's the green segment. The phosphate group, which is the yellowish segment here, and the nitrogen base over here. And there are actually 4 different ones. There's A, T, C, and G. Okay? So those are the nucleotides that make up DNA. The DNA forms a backbone, so the sugar and the phosphates connect together. The sugar part and the phosphate part connect to the next sugar and phosphate, and I'll show you what that looks like here in a minute. And then the nitrogen bases form the middle. So the middle part of DNA is like rungs of a ladder. So the G connects to the C, and the A connects to the T, and so on. And these bases, as they connect together, form the middle part of the DNA strand, and it ultimately looks like a spiraling ladder. So that's what the structure of DNA kind of looks like. There are categories for those nitrogen bases that we talked about. The first category are the purines, okay? And purines are labeled as A and G, okay? A and G are also typically double ringed, okay? So they kind of look like this. A pyrimidine is a little smaller, okay? Pyrimidines are classified as C and T, and they just look like this, all right? So pyrimidines being the smaller one and purines being the larger one, these pair together. Remember, A bonds with T and C bonds with G. And in making that bonding pattern, they form DNA. Now, Chargaff, who was another scientist, came along and discovered that the percentage of A is always equal to T, and the percentage of G is always equal to C. So in coming up with that, it was really solidified that, the knowledge was really solidified that purines bond with pyrimidines, or G bonds with C, and A bonds with T, because they're always really equal to each other. Okay? So that's the basic structure. Now we get to the last few scientists here. Rosalind Franklin was really an underappreciated scientist in her time, but did a lot of the work in coming up with the structure of DNA ultimately. She studied DNA. She used a technique that was known as X-ray diffraction, or she used X-ray crystallography to take these pictures of DNA. And what she ended up doing was shooting X-rays at crystals of DNA, and a lot of the pictures didn't turn out very well, but one of them really did, and that was the one here at the bottom. This picture shows a crossing pattern across the picture, and that kind of indicates that it was a helix or a twisting pattern. And that is what Watson and Crick used, who are the scientists that actually wrote the paper on DNA, discovered that DNA was a double helix. They used a lot of Rosalind Franklin's work to come up with this spiraling pattern of the double helix that DNA ultimately is. So the double helix looks like a twisted ladder. Both sides are held together by hydrogen bonds, and those hydrogen bonds exist between G and C and A and T. So it kind of looks like this, okay? Here's my DNA ladder on the left and the right-hand side. Here's my sugar and phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Same thing over here. And down the middle, we've got G connected to C and A connected to T. These are all hydrogen bonds, and that is what makes up the structure of DNA. So that is it for the structure of DNA. If you have any questions about this, please feel free to come and ask me outside of class, and we can go over this, or even in class, we can go over this together. Okay? Thanks for watching, and we'll see you next time.